Good afternoon, everybody. Thanks for joining the afternoon session of the Education Day. We are going to have a first great session on the challenges and opportunities for EV-based clinical applications. We have 5 speakers, sorry. I'm Clotilde Théry from Paris, France, and my co-chair is Lucia Languino from Thomas Jefferson University in Pennsylvania, and I give her the microphone now.
Thank you, Clotilde. Hi, everybody. I think there are still people coming in, but we do have to start. Oh, thank you. Can you hear me in the back? Can you wave? Yeah? Okay. So we're going to start now. Our first speaker is Dr. Mario Gimona. Dr. Gimona is from the Ludwig Boltzmann Institute for Nanovesical Precision Medicine. He is also the director of the GMP unit at Paracelsus Medical University in Salzburg. The title of his talk is GMP Grade EVs. And I'm glad that most of us miss the real definition of GMP, because including me, Mario, I thought that GMP was mostly about personnel. No, it's about facilities. So we learned something important today. Mario, looking forward to your talk.
Thank you. So it should be understood that any translational efforts that should bring any EV-rich preparation to the clinic must force and have a thorough understanding of GMP for a very simple reason. Anything that you intend to give to a patient has to be manufactured according to GMP standards. And that is not only restricted to products that are already on the market, but also to investigational products, and this is essentially what we all are dealing with. So the understanding of GMP is essential to progress beyond the stage of the bench and preclinical work towards the clinical, and I'm going to go into that in a second. So GMP, or good manufacturing practice, covers a lot of things that most people won't think about. It covers not only the manufacturing as such. Well, Bernd Giebel knows enough about GMP. You can leave now, thank you. But it's also about the personnel. You have to think about how to train personnel according to GMP, how your facilities and equipment are compliant to GMP standards, how you can start looking into quality control that includes the mechanism of action or critical quality attributes that I'm going to allude to in a second. How you control the process and the substance, and how do you do proper record keeping, and I will go into that in a second, why this is important. The interesting aspect of that is that GMP does not only apply to naive, unmodified EVs, but also to engineered EVs. It's essentially the same idea to have a quality control in place that is safe for the patient, and that's the final aspect of GMP that you have to consider. When we look into unmodified EVs, clearly that's MSC-derived EVs, it is evident that the idea of transmitting bioactive cargo, whatever it is, let it be RNA or protein or lipids, that can give rise to an interesting therapeutic that we can apply to help people overcome a certain injury situation or even diseases. Nevertheless, what we have to face is that any new drug, any new component that should go into the clinic requires clinical testing. And if you look into that picture, you find GMP right at the bottom of the intersection between preclinical and clinical stage, and that's important. So what this picture should tell you is that we need to have a certain congruence of GMP manufacturing and the translation into the clinic, and into the clinic translation means that there should be a congruence towards the indication that you try to actually deal with. What this also means is that GMP should really force you to think a little bit like manufacturers, and manufacturers think much different from people who do basic research work, and that's listed behind me. You have to think about items that are not necessarily coming to you easily. So the safety aspect has to be the predominant thinking, because the safety applies to the patient. It's not your own safety, it's the patient safety. And that means you have to look into product stability. That's something that we don't do at the bench regularly. Identity, and I will come to that, is a very, very critical issue. Potency is one of those challenges that we're facing, because how do we define potency if we don't really know what the mechanism of action should look like? And all of that then leads to the quality aspect that is the essence of GMP. And that means you have to have proper control over the process and the product. And the reason why you need all of that, in essence, is right here on the right. There is a hierarchical relationship between defining a mechanism of action and then defining so-called target product profile. So you need to clearly have an idea of what your product should look like in order to accept or dismiss the product before it goes into clinical testing. And only if you have these two items answered, you can then address the third one, and that is critical quality attributes. Simply speaking, that is, if I want to apply quality control, what should I be testing for? If I don't know what I'm testing for, how can I provide safety to the patient? Now, the downside of that is that when we look into EV-rich preparations are a sum of highly heterogeneous cell populations, no matter how you look at them, and poorly defined extracellular vesicles. So in the essence, all of this gives rise to a rather heterogeneous secretome fraction that you might fractionate in various ways, but it is not a defined medicinal product the way a synthetic drug is. And we have to acknowledge that. So GMP manufacturing always goes hand in hand with clean room manufacturing. And GMP manufacturing in the clean room is a very important prerequisite to really go into translation. But it does not solve any of the problems that I just portrayed. It can only help you to limit heterogeneity and give you a higher batch-to-batch, lower variability. This is what you want, because you can't define quality attributes based on highly heterogeneous batches. Now, quality control, however, will require many different assays. Starting with the cells themselves, if we go back to MSCs, well, they have to be MSCs at the time of harvest, not necessarily at the time of seeding them into a bioreactor. This is something you have to be clear about, because you have to show these, in essence, to the regulators. And there's a whole list of quality attributes that I listed here, and quality tests that you can do. But what we also have to realize and be very open about that is all of these assays give you numbers. They're not telling you much about the therapeutic activity or the potency of your preparation. And it's very, very difficult to really, really see that through the regulators. If you just say, well, but that's the numbers there are. They're not interested in numbers. They want to see that you do physical-chemical characterization. But this, in the end, is only numbers. So again, this hierarchical relationship between indication, biological activity, and mechanism of action is this very interesting triad, which, well, you can name it the good, the bad, and the ugly, if you wish. Because the problem that's lying behind is that when whatever you look at in terms of indication, and I just listed a number here, it must be quite clear that the reason behind all of these diseases that you should see here, I just didn't list them, well, they can't all have the same reason, the same underlying circumstances. So we can't treat all of these assays with the same EV preparation. We have to define a mechanism of action that is congruent with the indication that we're looking at, because we want to help the patients. And even more than that, if we look into this further, the mechanism of action is largely driven by the mechanism of interaction between the EVs with their targets. And most of us, of course, think it's the target cell, but very few of us have actually identified a target cell in vivo. And even more than that, it's not only these things that are important, but there are interactions that happen with non-cellular components like extracellular matrix, or we have components in the surrounding of the extracellular vesicles that have activities themselves that we don't need to deliver to the cell, but to the site of action. And that changes the way we have to think about defining a mechanism of action. Because first we have to see what kind of biological activities we have, and then describe them, to then be able to force ourselves to make proper quality attribute assays. And one of those that came up early, and I listed it on the top, so I added this. Sorry, I have to go back. So this is the classical situation, but I added a number 5 here, and that is looking into uptake-independent mechanisms, things that the EVs do themselves before they fuse or being taken up, and that's maybe equally important. And one of those items that has raised some interest over the last years is the membrane-bound CD73 5' ectonucleotidase that generates adenosine in the surrounding of the EVs. And this is an interesting aspect because we can, in fact, generate an assay to measure how much adenosine EV preparations can generate per particle. And I guess most of us still have the idea that one of the activities, the biological activities, and hopefully the therapeutic activity of EV-rich preparations is based on the fact that we modulate the immune system of the organism, so the immunomodulation term. But immunomodulation is based on what the immune cells actually do. And it is known that adenosine drives a lot of different things in different immune cells. And so there is a certain congruence and correlation between the activity of CD73 in vesicle-rich preparations and immunomodulatory and hopefully therapeutically positively acting effects. We have devised a very simple assay to actually measure the amount of adenosine generation per particle, which then in the long run can serve for us as a quality control measure. You can do this with a variety of different components that you find, but you have to make sure that you can validate that assay according to GMP. Otherwise, it will be less useful for further progress. Now, the interesting part and why the translation is still not where we want it to be, and this is the recent survey. There's only about 72 listed clinical trials that use EVs as an interventional substance. However, if you look closely into these, only 15 are really interventional. 49 are not even looking at direct intervention. And one of the reasons is that it is very, very difficult to do what I just tried to portray to you, to really answer and address all the questions that the manufacturer has to ask. And if you can't get beyond that stage of the manufacturer, you have a very hard time to move one step further, and that is moving into filing an IMPD or IND to the regulators. And this is just page 1 of the glossary of IMPD. I'm not trying to discourage anyone here. You can hate this, but I actually love that. It's terribly educational. If you read through that, you really have to ask yourself, can I answer all of these questions properly? Like mechanism of action, target product profile. Well, if not, and that's the downside, you should not start addressing regulators because they insist on answering these questions. You don't have to solve them, but you have to have an idea of what you want to tell them. And the simplest of all looks like this one. So we have quality, a quality section and a safety and efficacy section. And in one of those sections, there's this very simple line. So the proposed mechanism of action should be discussed. You don't have to solve it. It should be discussed. You have to have an idea. You can be wrong in phase 1. That's okay. Well, not brutally wrong, but somewhat wrong. But you have to do that, and for a very simple reason, because if you don't address the mechanism of action, all of those items come in. So how do I dose? What assays am I using? How do I define purity? What's potency? What's identity? If you don't have a mechanism of action, it's very, very poor to really run through that. So I would like to leave that on for a second. So the idea of GMP is not to really make life difficult for researchers. It is, and I hope that we can raise this understanding. It is primarily about the safety of the patient. And I think we all may agree that we need to make progress, and this field is enormously important, I think, to really make interesting therapeutic progress. But we have to be careful, because any mistake that we make, wherever we are not careful enough or too quick and not asking the right questions, could harm us all in that progress that we want to make. And the progress can be scientifically on one side, but it has to be paralleled by progress in the translational aspects. Yeah? So rather step back and go back to the original idea of your IMPD and look into that, what it really tells you. Again, it should help you to raise the right questions. Again, it's highly educative if you do that with students. Students like to, you can interpret things in numerous ways. But if you do IND or IMPD filing, you have to be very clear about the answers that you want to give to the regulators. And if you face the problem that you don't really know enough about what you're doing, then it might be wise to just step back and say, well, let's try to find out and solve any of the problems that are portrayed. So do we really know the state of our cells? Do we know enough about the loading? Do we know enough about targeting in the body? Do we know enough about how to administer the EVs properly? Have we calculated any side effects? And is it enough for us to understand what the literature says? Well, it's immunomodulation. What's our understanding of immunomodulation? Why am I picking on this? Well, those of us who are in the field for a while have come through the era of MSC therapy. And those, well, we know that. The original idea was wonderful, right? It's a stem cell, and the stem cell can go wherever there is an injury, differentiate into any cell that is damaged, and replace any damaged cell there is. Well, that didn't really surface, and we should learn from that. It's not that MSC-derived therapies were not according to GMP. No, that's only the manufacturing part. But there were certain aspects that were not addressed properly. And I think we're all lucky, and some of us are here because we are lucky, that nothing dramatic happened in MSC-based therapies. But we should try to avoid any of those going close to these near disasters with the EV therapies. So in summary, I think there is good reason to think that we can make progress towards translational field. So GMP-compliant manufacturing of EV-rich preparations is possible under GMP. It's hard, but it's possible. We know from a number of studies that there are satisfactory safety profiles in human trials, or at least in human applications, that have no gross side effects. That's a good start. This cannot be taken as a carte blanche to just do what you like. But there is evidence for it, which raises the potential to really eventually, in a few years, generate an off-the-shelf product that really can be given to people for certain indications, but not for everything. The downside of it is, and I think you spoke about that in the morning for a while, and it will continue throughout the day, we lack suitable reference material. We have no null references for biochemical assays. We have to develop those. And they have to be validated to GMP. Otherwise, they are not as good tools as we need them to be. The scalability issue drives us all. We need, depending on the indication, you have to be clear if you start going into a translational path, can we, or anybody that I know or hire, produce 100,000 doses per year? If not, you have to reconsider going forward with this particular indication. It could be problematic in the long run. And finally, I think we are all unclear about the mechanism of action and the biological activity of all the compounds. And that is highly problematic. So the upside is there's a lot for us to work on. So those of you who just entered the field, hey, there's an open prairie here, knock yourself out. But keep GMP in mind, and make sure that you understand GMP to the right degree, and that is, GMP is not voodoo. Yeah? This is just work, so don't get discouraged if you're continuously asked to generate more paper and do it again and answer the same question again. In the end, if we succeed and follow that translational path and end up in clinical studies, then it's all worth the trouble. With this, I would like to finish, and thank you for your attention.
Thank you, Dr. Gimona. We're ready now to take questions. Okay, I will start. So, Dr. Gimona, that's really a terrific presentation, very helpful. The simple question is, if you don't mind touching on the comparison between cell-based therapy and EV therapy, and what will be the advantage of moving into a GMP facility for EV therapy? Or could we combine? Could we have facility that will do both EV and CAR T cells therapy? Because that's what's happening, at least in America right now.
That's an interesting thing. I'm totally blank on CAR T. So that's a different scenario. The thing is, and I guess that's the advantage of EVs, that by definition we will make EVs an allogeneic therapeutic. CAR T is autologous. Most MSC therapies started as autologous, which has a GMP problem because that is every therapeutic approach is its own manufacturing process. It's like, people would love that, but it's like if your Ferraris are built to scale. But if any car that you buy has to be made especially for you and licensed especially for you, that just drives the costs through the wall. So I would not do that. So I think that's the biggest advantage of EVs, to really have a handle on something that is closer to an allogeneic therapeutic than anything else.
Thank you, Dr. Gimona. Yes, there is a question there on the right.
So thank you for the nice presentation.
Go on, I just need to hear that. Please,
Go ahead, please speak up.
Okay, so thank you for the nice presentation. So in case of MSC-derived EV, the mode of action is not so clear. So in such a case, how do you define the API and impurities? And how do you control the purity? Please share your thought.
Brilliant question. What is purity? What is an EV? I love that question because how do we define it? Is it the naked EV, so we purify it to death until there's nothing left on the membrane? Or is it anything that's attached to the outside? Call it corona, call it whatever you want. I think that's the hard part in the definition, and I don't think that we can address that properly because nobody knows really what's happening. But you can address this under GMP by saying, look, this is my manufacturing scheme, so I will have EVs that look like that. And then you start characterizing that particular product. And that gets you then closer to a mechanism of action. But the purity issue is an interesting thing, yeah. But I could not answer that. I would love to, but I plainly can't.
Okay, thank you.
Thank you. So if
There is one question coming. Yep. Oh, sorry. Oh,
no.
There is one question there.
Microphone, please.
Hi, really interesting talk. So I have a question. So alofisel, the only MSC approved therapy, was withdrawn actually in December. So what do you think that means for the MSC EV fields?
I could only speculate because I don't really know why. What was the reason for the retraction?
It didn't show efficacy superior to the placebo.
Yeah. Well, there's the difference between calling something purity or showing enough data to convince the regulators that this is the purity that you have and you can manufacture that constantly. And that's a pure GMP issue. Yeah? So in our case, I didn't have the time to go through that. I might go on Saturday. But we definitely do not have a preparation that you would call pure by any terms. But we can manufacture that particular purity again and again and again and again. So I have something that I can characterize and simply write down the numbers. And then the regulators say, okay, this is your level of purity. If you can then, out of that, carve out a mechanism of action, that's good enough, unless all the components that are in there are known and you know essentially what you're doing.
Dr. Gimona, we have one question here. We
thank you.
have two questions there on the right side.
Yeah, thank you. I think you showed a few of the challenges facing EV-based therapeutics for achieving GMP, but I was just curious, and maybe it's too broad because the field is so broad, but just given where EV research is today and where EV-based therapeutics are today, what would you say is the #1 challenge facing EV-based therapeutics for achieving GMP?
That's a twofold thing. I personally, because I'm a very simple mind, I go for unmodified naive EVs because we can calculate that much better. The bigger promise, definitely, and you will address that, I assume, will come from engineered ones. However, if you do that, you reduce the extracellular vesicles more or less to drug delivery vehicles. And the downside of that is that the loading and the targeting and the release of the cargo is still a large issue. So asking that question, I personally believe that the first ones to succeed are the unmodified MSC-derived EVs. And that should only set the baseline. That's my prediction. Like most times in my life, I may be brutally wrong.
Can I have one more question, Dr. Gimona, there?
So one key component of GMP is quality control. I think for you to release a batch, they need to meet your release criteria. But things as mentioned multiple times this morning, there are so many different modalities to characterize EV. So do you think that process of determining release criteria will risk being arbitrary? Thank you.
I think I tried to point that out, that what we're having now as EV preparations is highly heterogeneous. It's very, very ill-defined, and we can only poorly define it. Secondly, if we look at all the assays we're using, very few of them really tell us what we need to know. However, what we do under GMP is work according to the state of the art. We can't do more than that. Yes, we are encouraged to push the envelope and discover new things, but there's a certain standard, and the regulators know you will never be able to quality control a vesicle preparation like you can do with a synthetic drug. So the understanding is simply what are the limitations of GMP. It's not what it's asking us to do. The regulators know it themselves, and they know how experimental the whole thing is. So the most important thing to me at the moment still relies in the mechanism of action, because only if you have the mechanism of action, you might break it down to a molecule of action, which then allows you a correct dose finding. And I think these are the two items. As long as we can't do proper dose finding, we might get stuck.
Thank you.
The dose finding is even more important. Let me just finish that. If you're not clear about the indication, there are indications where you control the doses much better than in others.
Thank you, Dr. Gimona. Sorry, but the time we're running out of time. Thank you.
So thank you. And the next speaker is Dr. Samir Andaloussi from Karolinska Institute in Stockholm, Sweden. Please, Samir, he will tell us everything they have done for engineering EVs also for therapeutic purpose. Thank you.
Thank you very much, Clotilde, and thanks for the kind invitation. I'm pretty shocked to see how the field has grown. I last spoke in an educational session, I think that was in 2015, when I first met Bernd Giebel in Washington, D.C., and a lot has happened since. I think still, though, there are challenges that remains. So my talk today is going to be about EV engineering and how we can address certain challenges with using EVs for drug delivery. I will start with the easy engineering, meaning engineering the outside of an EV, to move to the more challenging engineering principles to load proteinaceous species into the vesicles. and finish off with the challenges, the most challenging part, which is loading functional RNAs. But before getting into EVs, there are some common problems or difficulties in our body that limits distribution of nanomedicines, which I qualify EVs as, like viruses. And the first obstacle we have is the endothelial barrier. And I feel that that is something that is quite largely overlooked when people talk about EV delivery and non-viral delivery in general, is that the endothelial lining of our blood vessels is very different depending on organ. And as you can see from this picture, organs like liver, spleen, bone marrow are fenestrated. So that means that if you have a nanoparticle or an EV that is 80 to 100 nanometers, that's small enough to pass those endothelial linings. Now, if we look at other organs like skeletal muscle or heart, they are not fenestrated. They have very tight capillaries. So it means that for a particle to reach the interior of a tissue, you need to cross that barrier. Viruses have evolved to transcytose and do that to a certain degree. The question is, has EVs? And I will get back to that, why I think this is a very important thing to consider, at least from a delivery point of view. Maybe not as much if we use native EVs that kind of kiss and run or acts in circulation. The other big barrier, which I think is more acknowledged nowadays, is endosomal escape. So once you're in a tissue, and you've been taken up by endocytosis in a cell, which is the mechanism that most EVs are being taken up through, the big limitation becomes the sequestration in endosomes, because the endosome is effectively outside of a cell. It's not actually inside a cell. It's captured from the outside and it sits inside. And for a cargo that is inside an EV, to reach the cytoplasm or the nucleus, it has two membranes to pass. Viruses have evolved to learn to do that, membrane fusion. They've developed viral glycoproteins to do that, so they can fuse and release their cargo. And I will come back to that also in my talk. So, what are the, let's say, what I see as pros and cons in using EVs to deliver biotherapeutics? Yeah, on the pro side is that you protect the RNA and protein cargos. It appears safe to repeat dose. I think in most studies, also in higher species, you can repeat dose and you can dose escalate and you see very, very little immune responses and tox responses, which is in vast contrast to most viral vectors or other synthetic nanoformulations. Delivery across barriers, I used to always have that as a point in my advantage talk about EVs. I'm putting a question mark because I'm not so sure anymore. Not without extensive engineering. The other thing is, of course, that I believe that they are potent surface signalosomes. There are a lot of goodies sitting on the surfaces, and you can enrich that also with engineering, and I think there are multiple examples of that in literature. So they can be engineered, and I think that's kind of important. So the cons, very fast plasma clearance, very similar to viruses. I will show some data on that. Limited extrahepatic delivery because of how our blood flows and how our vessels are built up, unless you have a disease. Poor loading of RNA, very limited payload release, and what I mean with that is that if you engineer a protein to sit in the interior of an EV, it has to be released. If it sits in a membrane scaffold, it has to be released. And I would dare to say that relatively insignificant endosomal escape. So there are some stuff to work on here to try to develop them as therapeutic delivery vehicles. So how do we engineer EVs? There are two ways. Either it's the endogenous loading, so it means that we genetically manipulate producer cells with an EV protein, so an EV sorting domain that we know enriches in EVs, either sits in the membrane or in the lumen, and we fuse that to our protein of interest. The other way to engineer EVs is what we typically call exogenous loading of cargo, and that means that we have our pre-isolated EVs, and we then, by different physical means, try to push our cargo into the lumen of these vesicles. So these are the two different approaches. So if we start with exogenous loading, and I will not talk as much about exogenous loading as endogenous loading. So there are different ways to get the cargo inside the pre-isolated EVs, and that is typically electroporation, sonication, or just co-incubation. If you have a lipid tag on an RNA, you incubate it with your EVs, hope that some goes to the inside, most stuck in the membrane. And then you have, like I said here, the membrane-associated loading. The advantage is, of course, that the efficiency, if it's small enough, the efficiency is relatively high. You can control dose, and it's rather quick compared to doing genetic engineering. The limitations, though, is that you really need very, very high concentrations of both your EVs and your therapeutic RNA, if it's a small RNA. And I would say so far in literature, it's mostly been small molecules that have been successfully loaded using this strategy. There are mixed successes with small RNAs, and I would say that loading large RNAs and protein therapeutics is basically impossible because of the size nature. So if we then move to genetic engineering, how can we achieve that? As I said, we have an EV sorting domain. We put our cargo, that could be a CRISPR-Cas9, it could be a Cree recombinase if we just want to look at activity. And these could be different. We can then use different EV anchors. We can have membrane-associated anchors that sticks to the membrane. We can have luminal proteins that normally sorts into EVs that we put as fusions. We can engineer the termini of tetraspanins so that we load our cargo of interest. We can stick it on single transmembrane domains. We can use multimerization domains to bring ligands together. There are examples of that. And we can use GPI anchors if we want to stick things on the surface of vesicles. So these are basically the toolbox that you can work with. And as you can appreciate, there are hundreds of proteins in each of these categories. So why do we engineer and what do we engineer? Either we can engineer just to study EV biology and biodistribution, and I will show some examples of that. But more importantly, we can engineer for therapeutic purposes. And that could be to alter PK, distribution. It could be to load luminal cargoes and promote endosomal release. And these are just some examples of different modalities that you could put on. You can have decoys or ligands displayed in different ways. I will show one example how we can put antibodies on EVs to target them. We can engineer for tissue tropism with single chain fragments, and we can also load the interior by putting them on those exosomal scaffolds. So, as I said before, pharmacokinetics and biodistribution of EVs, if we look at the average half-life in literature, if you do cargo-based detection, it's very short, somewhere between 2 minutes and 30 minutes, depending on EV source. And they're rapidly degraded in peripheral organs. The distribution is mostly to liver, spleen, and a little bit to lungs. Unless you have extensive aggregation, then you will see more in lungs. So really what this tells us is that you require surface engineering to enhance PK and really facilitate targeted delivery. And ways to do that include don't eat me signals like CD5547, albumin binding domains that I will show examples of. Putting antibodies on is another way to really try to steer the distribution. You're never going to avoid hepatic clearance. That's still going to be the predominant destination for injected EVs, at least intravenously. There are a whole slur. I will not go into the details, but there are different ways to label EVs if you want to study pharmacokinetics and how they distribute. And you can use fluorescent tags, either lipids that sticks into the membrane. The disadvantage with that is that they can fall off, so you don't know if you're tracing your EV or if you're tracing the lipid. You can also do recombinant fusion proteins, so you have fluorescent proteins on the inside. Then you have cargo-based detection, but the penetrance is limited, and you really need to do single-cell suspensions to look at the distribution, take out organs. What we do a lot is bioluminescent imaging, so we put luminescent enzymes on the inside that are very sensitive, but most of these also require that you take out tissues and then lyse them. And you can use radioactive tracers or contrast agents to study distribution. And this is basically a repeat. We can also look at this functionally, so if we can address functionality, not only where they go, that we do with luminescence and fluorescence, but you can also exploit fluorescence, but in recipient cells as a readout for successful delivery. And one such system is the Cree recombinase system that I will come back to. Another way is to use a CRISPR-Cas system reporter that when you have an edit or a recombination event, you get GFP expression. So you can probe for GFP in cells. So, as I said, these are just two examples. When we inject EVs, they go body-wide, but they go mostly to liver and spleen. And what you see is that they decay very fast. So, high uptake, and within minutes, it drops down. it's degraded. And we can follow this in real time also by using another luminescent reporter. What I don't have time to show, but what we've seen is that the reason for this very fast clearance is because most of these EVs that we inject, they bind to platelets and erythrocytes, then convey them to macrophage degradation in these peripheral organs. And this is not very surprising. You know, these are just two examples from the virus field, adenoviruses, how they are cleared, clearance kinetics, VLPs. They are cleared within minutes. They are cleared by LSEC cells in liver and by Kupffer cells, predominantly. And so the fact that we see similar kinetic clearance curves for EVs is not very surprising, I would say. I mean, the half-life differs a little bit between studies, but that can depend on how we label them. So how can we do to avoid this fast clearance? Yeah, there are a couple of different approaches. One that we've taken is to put albumin binding domains. So we engineer the extracellular loops of tetraspanins with 50 amino acids, same as bacteria used to bind albumin with high affinity. And by doing so, we can prolong half-life by about 20x in mice. Others have engineered EVs with CD47, CD55, and extended half-life. We, in my lab, don't see really robust extension of half-lives by don't-eat-me signal engineering. You can also put albumin binding domains synthetically by using lipid anchors, by using small molecules that bind albumin with high affinity and thereby extending half-life. Or you can pegylate the EVs, similar to how you do with synthetic lipid nanoparticles that are used clinically. I will skip that. So those are ways to extend half-life. Now, the other thing we also need to consider is how can we change if we want to target a specific tissue or tumors. So in this case, for example, we developed a system where we genetically engineer producer cells that instead of having an albumin binding domain, they have an FC binding domain. So the EVs that comes out on the right side here have an FC binding domain that will recognize any human IgG antibody FC portion. By doing so, we can plug and play and take any IgG and stick that on the surface of EVs. And when we do that, we can see robust tumor targeting in vivo in tumors that we typically don't see EVs accumulating in. And this is just an example of a PD-L1 positive metastatic melanoma tumor where if we inject non-engineered EVs or EVs engineered with an isotype control antibody, you see that there is basically no accumulation in tumor. Whereas if we look over time, when we use a PD-L1 targeted antibody, we start seeing accumulation in the tumors. Having that said, and this is important, if you look at the kinetic curves of clearance in blood, they overlap massively. So the clearance is pretty much the same. If we look at tissue distribution, what we do see is we have a little bit less going to liver, a little bit less in spleen, but overall the gross distribution is quite similar. So there is by no means solved. We probably need to combine these different strategies. We'll skip that and move over to protein loading, the more challenging part. Now we talked about how you can surface engineer to steer distribution. The other aspect, can we deliver what is in the inside? So this is an example of what we believe are the crucial components to successfully deliver protein therapeutics. You need your protein of interest, of course. Again, could be Cas9, could be a transcription factor, could be Cre recombinase. You need a smaller linker that can be cleaved because you tether this to a membrane-associated protein, in this case CD63. But what you also do need is something that makes these EVs fuse once they are in endosomes. And next slide will show that. You need cargo release and you need endosomal escape, because if you don't have both of those components, it doesn't matter how much EVs you're going to put on the recipient cell. And this is just to show what happens actually with super resolution microscopy. We've done this with a lot of different EV sources on a lot of different recipient cells. We see this phenotype that the more EVs you put on a limited number of recipient cells, what happens is that LAMP1 positive endosomes just start swelling. They grow, they begin to become bigger, and you can see in the yellow, you have green, they are really engulfing the EVs, but they stay in the lumen of these large endosomes. We don't see any escape events. And this is pretty much what this slide will show you. Here we have developed a system where we have an EV scaffold protein. We have a small cleavable protein called an intine. So that's a mini protein that gets self-spliced on a protein level when pH drops. So during EV biogenesis. And we use a viral fusogenic protein to induce endosomal escape. And then, of course, we have our payload, in this case, Cree recombinase, stuck with this intine linker to the exosomal protein. And if we do nothing, we just put tons of EVs on different reporter cells, Cree recombinase reporter cells, loaded either with Cree, CD63 Cree, so you know that you have a lot of Cree, an intine Cree or CD63 intine Cree, so you have the releasable system. There is no recombination whatsoever with any of those EVs. If we now do exactly the same experiment, but we put the fusogenic protein into the blend, we max out recombination in a dose-dependent manner. I didn't show that here, but this is now soon out. But basically it shows that you need two things. You need a fusogen, but you also need this releasable intine. This could also be a GAG if you work with a virus-like particle and not an EV. This is the equivalent to GAG-POL. You put the protease cleavage site in front of GAG. What you see is that if we don't have the intine, so if you use CD63 Cree and you add VSVG, you also don't get recombination because the Cree is stuck to the membrane of the vesicle. So you need that release in the lumen, and then you need that fusion to release your cargo. And we've used this successfully now together with Roos van der Broek's lab to induce robust both Cree recombination in the brain of mice when locally applied, and also with gene editors. Now for the last few minutes, I'm switching gears to RNA molecules. I realized that I've been talking way too slow or had too many slides, either way. So RNA is more tricky. Proteins we can get, depending on size, about 100 to maximum 200 copies. If we do luminal loading by surface engineering, we can get about 500 antibodies per EV with the best constructs. With RNA, natively, if you think about the size of an EV, you can only fit about 10,000 total RNA molecules, because of a total cell, you have 0.0001% the volume in an EV. So if you do the math and look in literature, if we look at the most abundant RNAs, like tRNAs and yRNAs, you have about 1 copy per EV. If you look at microRNAs, you probably have about 1 copy and maybe 100 EVs, depending on what microRNA you work with. mRNAs is getting less and less abundant. And this is important, because if you're going to deliver an mRNA and you only have 100 copies in 1 million EVs, you will need a hell of a lot of EVs to get a functional transfer. So you need engineering again. That's what we believe. And you can do that in different ways. You can have passive loading, just overexpress your RNA and hope that you get it into the EVs. You can do active loading by using different exomotifs or RNA that exploit native RNA binding proteins. Or you can do engineered sorting into EVs, meaning that you put an RNA binding domain on your EV scaffold and you put a recognition domain in the 3' UTR of your mRNA that you want to deliver. I will skip this, and this is actually what Joshua Leonard started doing now 10 years ago, where he, for the first time, fused MS2 RNA binding domain to LAMP2 or tetraspanins, and he then engineered the 3' end of the mRNA with the MS2 stem loop that is recognized by MS2, that then drags the RNA into the EVs. We've taken that one step further in my lab and started using human, because we want to avoid using as much viral or bacteriophage-derived products as possible. So we instead use a pomelio protein, PUF, that we use, and then we have this complementary PUF recognition domain put into the mRNA, in the 3' UTR of an mRNA. In this case, we did this for the OX40 ligand mRNA. And we then did, so we loaded, we could confirm that we can enhance loading by about 300-fold. We only have 10,000 intact copies per 1 million EVs. So when I say that we're really engineered, we're still at very, very, very low engineering degree compared to a lipid nanoparticle. And we inject these in mice, tumor-bearing mice, using this OX40 ligand engineered EVs. We could see that a lot of these mice live out, and they are completely responding, so they're tumor-free, and they're tumor-free now for 300 days. And this, we believe, is a combined action of mRNA loading and OX40 ligand protein display. So, I'm getting towards the end. So what are the challenges, in my view? And it's to find alternative ways to mitigate macrophage uptake and fast plasma clearance. And we may need to consider exogenous modifications, because you can put more functionalities in if you use, for example, lipid tags or click chemistry. Protein loading is sufficient with existing scaffolds, I would dare to say. We can get good editing levels. But loading enough intact RNA is a challenge, especially in the context of mRNA. We need to identify non-viral protein components that can perform endosomal escape. I think from an immunology point of view, repeat dosing with a viral fusogenic protein is probably going to be problematic. And then it's the question whether we should go local or systemic, because what I didn't say here is really the reason we focus a lot on local applications of EVs for drug delivery is because of solubility reasons. Because if you think that you don't get enough payload per vesicle, you need to use a lot of vesicles. And in the end, you run into viscosity challenges. Locally, that doesn't happen, but systemically, if you use it systemic use, that becomes a problem. So the future direction, I personally think that we will see more surface engineering, maybe combining MSC EVs with other bioactive proteins stuck on the surface of the EVs. I think for enhancing luminal loading, we may have to consider multiple scaffolds, not using single scaffolds for loading EVs. I personally would opt to go for local applications, generally, therapeutic local applications, gene editing. I would avoid systemic, at least if we think about delivering to heart, spleen, and bone marrow, because lipid nanoparticles, and in particular emerging targeted lipid nanoparticles, are doing a really great job at that. And I think we need to be mindful about that there are other things than EVs that you can use for delivery. We need to identify means to selectively capture engineered EVs only. That's not easy, especially if you're engineering the interior. And I think in cancer immunotherapy, gene editing, vaccination, I think there is a lot of scope and work to do with EVs. And with that, I want to thank you all.
Thank you very much, Samir, for this great presentation. We have time maybe for one quick question. We are a bit over time, but please come to the microphone if you have one question. Well, I don't see anyone. Well, okay, Bernd.
Yeah, Samir, thank you very much. Excellent as always. So I have a question. Many in the audience or in the field prepare EVs and analyze the content of RNAs and predict models that they are delivered and think about the mechanism of action via RNAs. So what do you think of that? How should we deal with such studies in the future, knowing what you have seen now?
Yeah, I think it's a tricky one, right? I mean, if someone would tell me that everything is mediated by one individual RNA from a non-engineered cell, I think it's a numbers game. I just find it very unlikely because it would assume that every EV is taken up and that you have 100% endosomal escape. We've done a lot of calculations on this, what is reasonable, and if you have attomolar amounts in circulation, the question is how much can that do? But there can be combined effects. But I think especially in these inflammation models and what you work with, Bernd, is that you usually see very fast responses with MSC EVs, right? You see that you get this anti-inflammatory effect is quite rapid. And if that was RNA mediated, I just feel like kinetically it should take a little longer. But I'm not going to say that it doesn't work. But I think it's more challenging.
Yeah, there is another question here on the right.
This is very interesting presentation, sir. How do you separate the engineered EVs and then the naive EVs? Loaded EVs and then the...
Sorry,
How do you separate the loaded EVs and then the unloaded EVs? Loaded extracellular vesicles and unloaded extracellular vesicles.
Okay, so yeah, we don't. The answer is we don't. We do not separate loaded from non-loaded. We've tried to do proxy quantifications of how many are loaded, but we don't, unfortunately, and this is a problem.
What is the ratio of guide RNA loading in the case of protein, Cas9 protein loading in the engineered EVs? Like,
I think I'm also got 10 years older. I don't hear. Say again.
Yeah. No. As of my knowledge, whatever I saw is the guide RNA loading is very much challenging in case of the Cas9 editing. So how you are facing that one?
Yeah, I didn't include that, but that's actually something that we work mostly with in my lab. So I'm happy to, guide RNA loading is the limitation, 100%, not Cas9. Okay. Loading enough Cas9 is easy. you need to have the right structures of your guide RNA. You need to make it stable enough. We don't have, right now, my lab don't have a very good active loading approach for guide RNA, but we have a pretty good idea of how we can design the scaffolds to enhance the loading. Yeah.
One more question is, I have seen your 2 articles, both with Cas9 protein loading and then the Cas9 mRNA loading. Like in mRNA loading, how are you loading the guide RNA? So,
yeah.
I'm
So with the EVs, I know that there are in literature Cas9 mRNA loading. We have not been successful doing that. It's a very large mRNA. If you think about the size and the size of an EV, yeah.
Thank you, sir.
We have one last question in the middle.
Thank you. It was a great talk. I just have 2 quick questions. The first one is I quickly saw the use of mini-osmotic pumps. So I was wondering, could you talk a bit about the choice of the dose, the administration route? Yeah. And another question is, what do you think about the blood-brain barrier for naive EVs versus engineered ones?
Yeah, so I think the reason we use osmotic mini-pumps is exactly the reason for solubility. We need chronic release over time and not run into solubility problems. If we do a bolus injection, we get about 2% editing in the brain. If we use a mini-pump approach, we get higher than 40% editing in the brain. So that's the reason, and it can be clinically translated. Mm-hmm. What was your second question?
The blood-brain barrier.
Yeah, so that's of course how when I started working with EVs, that was the promise that was sold, that it's going to deliver everything to brain, turned out to be a little bit more challenging. Okay. Maybe when there is a disease, it's different, and also distribution. I didn't have time to show, but of course, PK is massively changed when there is inflammation, for example, or you have disease onset. This is all in wild type, non-inflammatory conditions.
Oh, okay. Thank you.
Thank you. So thank you very much for the presentation, for the questions, and we'll stop here.
Our next speaker is Carlos Salomon. Carlos is from the University of Queensland, Brisbane, in Australia. And he will tell us about EV biomarker discovery, validation, and translation. Thank you, Carlos.
Thank you, Lucia. Everyone can hear me. All right, so I'm gonna time myself. I have the timing there. All right, cool. So thank you to the organizer for the opportunity to share a little bit our experience in biomarker discovery. And also thank you, Samir, because I thought I'm gonna be the only one go over time, but no. So thank you for that, all right? So this is an education session, right? So hopefully this is gonna be some of the aspects that we're gonna be discussing today, including understanding the unique advantage of EVs as a biomarker platforms, differentiate between discovery-driven and clinical-orientated EV biomarker research. So what is our main goal when we design our biomarker studies, right? Recognize the key phases and challenges for EV biomarkers. Understand also the regulatory processes, right? How you can actually deliver your biomarker to the market, right? And then apply the criteria for clinical implementation of EV biomarkers. So we're gonna start with something very simple about what is actually the definition of a biomarker. So according to the FDA, a biomarker is a defined characteristic that is measured as an indicator of a normal biological processes, or can be also a pathogenic processes, or response to exposure of intervention, including therapeutic intervention. So one of the key aspects of this definition is a biomarker must be objectively measured. So you need to be able to quantify this biomarker and have to reflect either a pathogenic process or a normal process as well, right? There are different types of biomarkers. So obviously are diagnostic biomarker, which is identify the presence or no of a specific condition, prognostic biomarkers, predictive monitoring, which is actually EVs are quite attractive because it reflects the metabolic state of the originating cells. So we also have heard about they can be a good real-time monitoring system of disease progression. So if you are in the biomarker business like me, it's very, very difficult to actually deliver something to the market. And this is not only because you are, it's because you are working in EV biomarker. That's the difficult thing. No, actually, it's because you are actually trying to commercialize a biomarker. So this is a very interesting commentary that actually is almost 15 years old, but applied for 2025 as well. So it's just a few proportion, it's just a few percentage of all the biomarkers that we propose in the literature finally end up as a product. So you can see that it's actually less than 1%, which is make this quite difficult, right? So what are the main challenges for commercialization of biomarkers? And this is in general for biomarkers, not only for EVs, right? One is about lack of standardization, a small sample size, and this is something that we observe quite often in the literature as well. So we're trying to identify biomarkers for a specific cancer when the incidence of that cancer in your population is 50%, which is, it's not real compared to what is the incidence of cancer when you're trying to develop a screening test, for example. Overfitting the data, especially in your discovery approach. This is something very common mistake, right? So one of the things about biomarker discovery is whatever you discover need to be tested in a completely independent set of data. Obviously, they are limited in terms of the reproducibility and regulatory and clinical hurdle as well, and lack of the clinical utility as well. So keep in mind that to be able to develop a commercial viable product, that need to address a specific clinical gap as well. So why EVs are quite kind of attractive to trying to address all this stuff? One of the things is because they are, obviously, you can identify this in different biological fluids, for example, plasma and serum. And they contain information, and that information actually is a representation of the metabolic state of the originating cell. So it's just the basic concept about liquid biopsy. So can you actually track the progression of a specific disease or even the presence of the disease through this extracellular vesicle? And how you do it? You're trying to identify biomarkers inside of this or attached to this extracellular vesicle. How representative are these biomarkers and how specific are these biomarkers for the disease is something that also needs to be emphasized as well. So how can EVs address the challenges in biomarkers? So in terms of the low reproducibility, EVs actually are quite stable in circulation. We all use samples that have been stored for many years in different biobanks, and then you still are able to identify different biomarkers inside of these EVs. Sample degradation is something that also EVs can potentially address because the biomarkers inside of this extracellular vesicle are protected and are less susceptible to degradation as well, right? No invasive in terms of the sample required, so you can minimal invasive or no invasive at all if you use biomarker, for example, in urine, saliva, or even you can identify biomarker in blood as well, right? So probably this is one of the first message that I want to emphasize in my talk, right? And this is a decision that you have to make when you are working in your biomarker studies. So what is your goal with your biomarkers, right? Are we designing a study to publish a paper? So this is your end goal, just to publish a nice study? Or are we setting out to deliver a clinical useful biomarker that actually can improve patient care? Because this is two aspects that are common, it's a common mistake in all the biomarker studies. If you're planning to develop a biomarker, and hopefully an EV biomarker that potentially will reach the clinic, you need to be interacting with the biotech companies probably more often as well. So what I'm going to be doing in the next 13 minutes is I'm going to present some examples about what are the key advantages to use EVs as a biomarker, right? And the first one is about the reflecting of the cell of origin, which is quite interesting. This is a very interesting study published by Andreas Moller group a couple of years ago, in which they identified that when you isolate EVs from two different breast cancer cell lines, the difference that you observe on the cells are reflecting in the EVs, right? So this is the concept about liquid biopsy that make EVs quite attractive as well. This is something that also we pursued in my lab. This is something that we published recently in which we identified that placental alkaline phosphatase, which is a placental marker, which is associated with placental state, which is very important for a healthy pregnancy, you can track actually the metabolic state of the placenta if you quantify placental alkaline phosphatase in circulation. And you can see that for this particular discovery project, we have a cohort of patients around 200 pregnant women that we collect blood sample around 11 to 13 weeks of gestation. And we separate the data. We use half of the patient was used for training and discovery, and the other half was completely blind for the training. I was used for a tested of the data. So then how we actually evaluate that we have a good biomarker? It's not about up and downs in terms of the abundance of this particular biomarker. It's about sensitivity and specificity. Each of our biomarkers is able to identify the true positive in your cohort. If this biomarker is able to identify the true negative in your cohort. So in this case, women without risk to develop complication of pregnancy. And I suggest to do at least 3 basic analyses. One, obviously, is the analysis that we perform all the time in our biomarker analysis, which is about the ROC curve, which is a balance between which one can give you the best sensitivity and specificity. You can see the true positive ratio and the false positive ratio, right? Then there are 2 main analyses that also I suggest to perform, which is the recall and the calibration, just to determine how good is your biomarker to identify the true positive, and how good is your biomarker to identify the true negative in your population. The other thing quite interesting about this particular study, you can see 2 lines here that is in red and blue. So the blue one is the training, and the red one is the test, which is completely independent. So you can see that we are not overfitting the data, which is one of the main problems in biomarkers, right? Number 2 is they're present in different biofluid. And this is something that I'm pretty sure have been discussed through the day. So the EVs can be identified in blood, in saliva, in urine. So they're very accessible, and the content is protected, right? Stability in circulation. What is the advantage to use actually EVs versus just soluble biomarkers? And this is something that we need to ask ourselves when we run our biomarker study. This is a very nice example of that in the context of colorectal cancer. So you can see that initially they just measured this CEA, which is a carcinoembryogenic antigen associated with colorectal cancer. So you can see the area under the curve is around 0.8. This is the soluble antigen. If you measure in the EVs or exosome in this case, you can see you have an improvement in sensitivity of around 13%, increase in specificity around 38%, and overall the increase in the correct classification around 34%. So you are demonstrating a specific advantage to use this extracellular vesicles as a biomarker compared to just soluble proteins. The other thing quite important about EVs is scalability. If you're planning to develop a biomarker test based in EVs, you need to be able to run over 200,000 tests per year to be commercially feasible, right? So, and for that, you need to understand how you're going to isolate your EVs as well, right? So in terms of the biomarker, it's become very popular, the magnetic bead isolation system, because you can scale up this quite easily. And this is important depending on the intent of use of your biomarker, right? So keep in mind this needs to be compatible with pathology workflow, right? To run up to 200,000 samples per year, right? This is another nice example of a high throughput platform in which this author developed a droplet digital assay in which they measure PD-L1 and CD81, and you can see in the figure D, actually the correlation even much better than a commercial ELISA for the same biomarkers, right? So to effectively leverage the EVs as diagnostic, it requires the identification of the disease-specific biomarker, but also you need a rapid, reproducible, and scalable sample processing, which is compatible with pathology lab, right? The other characteristic about EVs that is attractive for biomarker is because the content seems to be protected, so it's more stable for your biomarker analysis, right? And this is one of the typical studies that was run by Andy Hill many years ago, in which they demonstrate that the macroRNA content in extracellular vesicle is protected against degradation. The other thing is the EVs contain a lot of information. You have protein, you have lipids. Actually, the majority of the content of EVs are lipids, right? And somehow this have not been well studied up to date. DNA, microRNA, etc. And this is a very nice study published by the David Leiden group a couple of years ago, in which they demonstrate that when we analyze protein analysis in different cancers in the tissue, they can separate actually the cancer with over 90% specificity and sensitivity. If they track the EVs coming from this tissue, actually sensitivity and specificity is around 90% as well. So biomarker analysis is not about up or down regulated of specific molecules. It's about can you reach the sensitivity and specificity that is required to actually develop a product, right? So keep in mind, sensitivity is about the true positive. Are we able to identify the subject of the person that have actually the disease in your cohort? Specificity is about your true negative, right? So I'm going to give you an example because ovarian cancer is one of the areas that we work, but if you work in other cancer, you can apply this for other cancers as well, right? So ovarian cancer actually is a low prevalence disease, right? So usually it's around 1 in 2,500. So the requirement actually to develop a test for early detection of ovarian cancer or detection of ovarian cancer that can be applied to the general population need to be at least 98% specificity, right? And the reason for that is, for example, you want to screen 10,000 women. Actually, women with the disease is going to be just 4 because you have 1 in 2,500, right? So 10,000 women, you might have only 4 cases, right? So a test with 90% specificity, that means that we'll identify 1,000 women with the positive disease, actually they don't have it, right? So that means that is 254 positive for each of the true positive cases, which is actually a problem. And you can see that false positive is a main issue, especially in the screening test, right? And that will affect specifically your positive predicted values, right? This is another example. For example, if you have a specificity of 98% and sensitivity of 80%, the positive predicted value will be around 6 to 10% in the general population. So imagine that you screen 100,000 women, right? So according to the incidence of ovarian cancer, in 100,000 women, it's going to be around 40 cases, right? With a sensitivity of 80%, you're going to be able to identify 32 of that 40. With a specificity of 98%, which is a lot of specificity, right? You're going to have 2% actually false positive. This is almost 2,000 women. And that means that the positive predictive value of this is going to be 1.6%, right? So this is the stuff that you have to keep in mind if you are planning to actually develop a biomarker project that actually you want to develop a product that will actually reach the clinic. The other message is about you need to understand what will be your commercialization pathway. And that also depends on where you are in the world, right? If you are in the US, also you have to interact with the FDA. If you are in Australia, the TGA, etc. So this is very important that when you approach this design of the biomarker project, just make sure what is your end goal on this. This is a nice example. You can find it online as well about what is the clinical need for the biomarker that you are developing. What is the commercial viability of this, right? So you need to address specific gap, and that need to achieve a specificity and sensitivity for your biomarkers. But hopefully it's a line at the end of the tunnel, right? And this is 2 biomarker tests that received some breakthrough designation from the FDA. One is for the ExoDx for prostate cancer, and this is another company in the US, which is the Mercy Halo for ovarian cancer. So there are no FDA approval yet, but at least some of these companies are actually trying to deliver this to the market as well, to actually help people at the end of the day, right? So some of conclusion and take-home messages. So the first one is clarify the goal, right? Are we designing a study just to publish a paper, which is valid, right? Or we are developing something that you can reach the market and help people, right? Recognize the unique advantage to use extracellular vesicle. Super important in your biomarker analysis. What you need to do is check sensitivity and specificity using cross-validated model. This is super important as well. Otherwise, what you're doing is just overfitting your data, and then if you spend time and funding to trying to validate this in an independent cohort of patients, it's more likely that this is not going to work, right? Make sure that your validation studies, you use log models. Otherwise, it's just another training. This is super important, I put in red, which is engage with, if you're planning to actually deliver a product, trying to engage with your commercialization teams as soon as possible. Connect with people in your institution. Probably over 95% of us, they're academics, so just talk with the people in your institution. And talk with the industry as well. So I think it will be important for us to be more interactive with the industry because they know what is the requirement actually to deliver and develop a feasible product at the end of the day, right? Understand the regulatory and the market landscape, so that will help you for your research as well. I know that this is going to be online in some point, so I put just some questions here. If you want to test yourself, how are you in terms of the biomarker field, all right? Just to finalize, ISEF actually is making a lot of effort to trying to understand the landscape for biomarkers, and this is something that we did last year, and we're planning to deliver, hopefully, the summary of the report as a paper in the next few months, so be aware. Also, we have an inter-society group between ISEF and the European Liquid Biopsy Society, so if you are interested in biomarker, please reach out. You're going to find Katerina and Julie and myself around during the whole meeting, so if you are interested, please talk to us. I have 30 seconds. If you have any questions, please reach out. I'm always available. Thank you.
Thank you, Dr. Salomon. Now this presentation is open for discussion. I'm going to start, Carlos, while people come to the microphone. Oh, there is one there already. Excellent. Okay, you're going to start?
Thank you very much for this very insightful discussion of biomarkers, EV biomarkers. I'm trying to better understand or perhaps clarify when you discuss the origin of improved sensitivity, especially, and specificity of EVs as better biomarkers. Is it because EV on their own right, so to speak, have improved profiles of biomarkers, or is the fact that it's actually concentrate very effectively protein biomarkers, and that allows you to improve sensitivity by virtue? Essentially, it's kind of internal pre-concentrator, and it allows you to improve your standard workflows as protein biomarkers. So kind of trying to differentiate between those two.
Yeah, so thank you for the question. I think the improved sensitivity and specificity when you analyze EV biomarker versus soluble biomarker is because the EVs will have a better representation of the originating cells. So for example, you're looking for a specific type of cancer and you're able to identify markers in the EVs that are coming from the tumor cells, that actually will help to identify the true positive and the true negative, right? Okay. I think probably that will be one of the aspects. The other thing is about stability and protection, right? Mm-hmm. So I think that also will help with your sensitivity and specificity.
Oh, great. And follow up on that. So do you then envision an adaptation or modification of your standard sample prep technology that's being used for diagnostic applications, specifically targeting EVs as the target system rather than soluble biomarkers? Because there is an interesting design game to be played that can take advantage of it.
Yeah, no, absolutely. I think what we need to define, and I get this question all the time, right? So I develop biomarkers which you can quantify in blood, in plasma, or in serum, right? So one of the questions I get all the time is, do you really need to isolate EVs to quantify this biomarker, or you can do it in plasma or serum, right? And yeah. I show some of the examples that if you do it in EVs, your sensitivity, specificity, and overall classification efficiency is much better than the soluble biomarkers, right? To answer that question, probably for a specific project, you have to do probably a similar analysis. Can you actually deliver a better classification efficiency when you quantify this biomarker in EVs rather than to insoluble form in plasma serum?
All right, great. Thank you so much. Very insightful.
No worries.
Carlos, I have a question. Yep, so I don't see other, but so actually I had two questions. One, if you would like to mention Carlos, Jon's Skog success, if we want to say that way, of identify biomarkers in the urine for prostate cancer, urine EV for prostate cancer. In that case, Medicare is also in America supporting the use of that test. So in that particular case, he's using 3 different RNA as a signature. So I'm wondering, when we talk about biomarker, should we think about a combination of more than one RNA, more than one protein, or are we looking at one specifically? Just a little comment.
Yeah, no, absolutely. That's a great question. I think one of the advantages to use EVs that you have the EVs, you have many different biomarkers inside of this extracellular vesicle. So that will help you with a combination of not 3, 4, or maybe 5 different combinations of biomarkers that you can develop a classification model and then look at the algorithm that can give you a good predictive performance, right, or diagnostic performance. So I think that is one of the advantages of EVs. Rather than to just measure a single biomarker, you can have a combination of them to trying to achieve the sensitivity and specificity that is required for a specific disease, right?
And then the second one, is there any FDA approved? Is it really required? Is an FDA approval really required for those type of screening tests? Because you probably will end up having more than one test in that for cancer.
Yeah, no, absolutely. You need the approval. So it's a couple of things, right? So you can have a LDT, which is essentially a laboratory developing test, in which it's just one lab is allowed to perform the test. But if you think about this from the commercial point of view, this is not commercially viable, right? So to be able to reach as many people as possible, you need to be able to develop a test that can be run in a hospital or in a pathology lab. And that needs to be in different hospitals, not only in your city, but around the world, right? So you need to have a specific approval for that from the FDA to be able to do it. The other thing that is important is about reimbursement, right? So how you can develop a test that is good enough to improve survival, then you can convince the government to be including Medicare or whatever health system that you have. And for that, for the cancer field at least, you need to demonstrate that not only your biomarker is able to identify the disease, but also that identification leads to a better survival of these patients.
Thank you, Carlos. Thank you.
Thank you.
Thank you. So our next speaker is Dr. Shannon Stott from Mass General Hospital in Boston, USA, and she will tell us about microfluidics and other emerging technologies in the EV field. Thank you very much.
Wonderful. Well, thank you very much to the organizers for this opportunity to speaking with all of you today. For anyone that knows me, microfluidics is something that I'm very passionate about, and it's really wonderful to be able to give you a little bit of background on microfluidics and how it can impact the EV field. I think opportunity is one of the big words of this session, and I hope that this will maybe engage a few more of you in microfluidics. So I figured to start with an introduction to microfluidics, since not everyone is familiar with it. I'd show you some different microfluidic devices. So this is a device that we designed in my laboratory. So I'm a mechanical engineer, and I'm based in a hospital out in Boston. And this is a device that we were using that there's multiple tubes going into one sample. So some of the tubes contain oil, some contain water, some contain small particles, as well as fluorescent dyes. And we use all of these tubes together with a variety of pumps so that we have very precise control of things as they move through the device. Some of the things that this device likes to do, I'm going to see if the video starts. Oh, my videos aren't working. Let's see, they were working in the speaker ready room. I don't know if, because this is, oh, here we go. It's just slow. So this is a, thank you. You just had to walk over to the podium. So this is a droplet-based microfluidic device, and what we do is use a fluidic force called inertial force for particles going through, and so that we can actually organize and line up particles as they're going through the microfluidic channel, and then it's pinched off by 2 oil streams so that you have a single particle inside an individual droplet. So this is something that was observed under a high-speed camera where these images were taken at 64,000 frames per second and then slowed down. But this is to highlight that you can actually do things pretty fast even though our devices are small. Microfluidics can also be quite simple. This is a very straightforward microfluidic device. You don't need pumps. You can just pipette in, you can use these small chambers. Microfluidic devices can even be made out of shrinky-dink material. So I don't know if anyone is familiar with Michelle Kine's work where she can take and pattern on shrinky-dink material, pop it into a toaster oven and shrink it down so that anyone, including my children, could make microfluidic devices if they desire. These are wonderful for resource-limited environments. Here's another example of a microfluidic device. This one is made out of an elastomer called PDMS, which is traditionally how research labs start off making their microfluidic devices, where they'll have an idea for a design, they'll create that design in a clean room, and then they'll pattern it on a wafer where you can then produce these devices one after the other, and then use them and then iterate on that design. For us, we can have an idea for a design and we can start testing it within about 48 hours, which I think is pretty exciting as an engineer for being able to go from a concept to start testing. But in reality, there's lots of complicating things that go into those devices. So even though we can make the base device pretty quickly, we often have to have lots of fluidic channels that go through, and this is the part where microfluidics can get far more complicated than it seems at the surface. So if I were to define microfluidics, I'd say that lots of different disciplines need to come together to make our devices work. We usually, though, are doing all of this to manipulate and control small, but I'd also say large volumes of fluid using these tiny channels. So usually our channels are anywhere from 10 to 100 microns, but even that definition can be broad on both ends. It's kind of like EVs. We can think about the really tiny ones and also the really large ones. Originally, when microfluidics started, it was using very small volumes for things that were going through, sometimes just a droplet of blood. But some of my colleagues at Mass General just created a microfluidic device that processes 2 liters of fluid within an hour. So you can go through all of these different scales when you're thinking about how can we control these individual particles as they're going through. It also involves the science and understanding fluid behavior, and fluid can be anything. Air is a fluid that we can manipulate inside microfluidics, as well as more traditional fluids like blood or plasma or cell culture media. And then we want to be able to design and fabricate these devices. So this is another area of opportunity and exploration, particularly where we have 3D printing now, where we can create and print all sorts of new 3-dimensional spaces. When I'm thinking about microfluidics and EVs, we want to think about all of those fluid behaviors that are driven by viscosity, surface tension, and capillary action. I don't want to get too engineering-like in my talk today, but often it's in the Stokes regime when we're thinking about fluid forces and fields, and so the Reynolds number is far less than 1. I just say that because most of the fluid dynamics field originally was for Reynolds numbers that were much, much higher. So this really creates an exciting time in our field where we can think about how do particles move when we're in this region of fluid flow, that also creates even more opportunities when we think of things on the size of extracellular vesicles. I also spend a good portion of my time thinking about material science. If you think about it, pushing blood through a teeny tiny space, you're going to activate a bunch of things. And so material science and how you coat those devices and the types of chemistry that you use inside them is also critical to think about. Chemical analysis is also something that can be done. It's not just for isolating out EVs, but it can also be for our analysis. We can do lots of mixing in very precise controlled ways inside these devices that can help with our analysis. And of course, as I was showing you, these devices can be very simple. Paper microfluidics is another area. Many of you have probably done a rapid COVID test. Any type of laminar flow assay is also considered to be part of microfluidics. So when you think about the benefits, one of the things that's really a major strength is that you can, if needed, use very small fluids. One of my more exciting projects that we're doing in the lab, we're looking at pediatric cancers, where sometimes we have children as young as 9 months old that are coming, and when we're taking their blood, we only want to use about 100 microliters of plasma from these patients because they simply don't have much to give. There's also the possibility for single EV manipulation. We're very frequently doing single cell manipulation with microfluidics, and now more and more groups are expanding to do single EV. You can also do multiple different processes on one chip. Originally, there was that term lab on a chip, but now we're doing particle on a drop, and this is where I see more and more EV work happening. And then you can also have low cost for many of these fluids, and again, it depends on how complicated it gets. So there are also some disadvantages for using microfluidics. Fabrication can be complex. As much as I say, oh, we can create a device in 48 hours, that's with a PhD in fabrication and lots of experience being able to go in. When someone joins my lab, it usually takes about 6 months before they're able to be trained and to be able to produce these devices. And while the devices can be simple, the tools that we need to drive them can be expensive. I can say that my chip costs $10, but you need to buy a $10,000 pump to be able to run it. So there's all these trade-offs in the early stage of using microfluidics, but we do have commercial products that are using microfluidic devices that with scale have gone down far less in price. For many of you who drive cars, your airbag is controlled inside with a microfluidic sensor. And for those of you that analyze RNA, digital droplet PCR also has microfluidic devices inside to create those droplets that are encapsulating our RNA. Operating devices can be tricky. Dr. Sara Vega, somewhere in this room, she uses all of our microfluidic devices, and she'll tell you those bubbles that happen, they really were very frustrating for her at the beginning when she was learning how to use them, but she's now a pro. I will say she's a pro in using them. And then I think the other thing that's that challenge is that all of the physics were based initially on cells when we're thinking about doing single particle manipulation. And it really creates this opportunity for thinking about using EVs in microfluidics. So to give you a little bit of my particular interest, so this is Dr. Alessandra Moore, who's now a surgeon out at Stanford, using one of the microfluidic devices inside our clean room. So like the GMP processes that we heard about earlier, we have to operate in a very clean environment when we're making our devices, because even if you just have a skin cell fall into one of your devices, it can clog it. But we're able to do and make these devices because we really want to help cancer patients and be able to create technologies that can be translated to the CLIA laboratory. So the Carlos's talk on biomarkers, I think, for me resonated so deeply because I was just nodding along of like absolutely everything that you're saying, and we believe that microfluidics may help bring some of that rigor. There's different types of microfluidic devices that can be used. One is for isolation. This is positive selection is one that typically people start off with, and this is also used for some of our imaging strategies that many of our vendors and companies use that have microfluidic devices, is where you say, I know something that should be on the surface of the extracellular vesicle, whether it's a protein or an antigen, and I'm going to coat the surface of that device so that it can capture that particular target of interest. There can also be negative selection, where you say, I don't want to presume what's actually going to be on the surface of my extracellular vesicle, but I know what I don't want. I want to remove all of the other components, and say in the case of whole blood, that I want to be able to take away. So I want to remove the red blood cells, I want to remove the platelets, proteins, and other unwanted cells. So this is a microfluidic device that I was involved in. It's 3 different microfluidic devices in one. In the interest of time, I can't explain all of the different things, but we do a microfluidic buffy coat generator. We also use that inertial focusing to line up cells as they're going through so that we can isolate out cancer cells from blood. One of the negatives, though, taking this negative selection approach, is that often the purity is reduced, where you have more contaminants inside there because you're not able to isolate out that individual. But it does allow for some pretty exciting research opportunities, as well as in the clinic. But for that device, I just want to show you what it looks like. So these devices are actually produced in Germany. So they're using the old DVD technology, which is why it looks like a compact disc. But if you look at one particular area, I think I have here. So there's over 400,000 microfluidic parts on this device. And so if I just going to say, in the upper right-hand corner, that inset to the right is just 1/1000th of the device. So there's lots of individual components that are there, and it takes a lot of engineering just to get these devices right. But the end results can be pretty impressive. So this is a movie where we have the exit of the device, and so I'll see if I can get this movie to play. There's been a bit of a delay, but on the bottom what we have are white blood cells that are coated with magnetic beads that have been removed from the device. And the device is flowing, and this is slowed down at 1/1000th of a speed. And I think there's just too much of a delay on the video, so I'll just show. At the end, it's very impressive. You have your cancer cells circulating out that are going through. So when we think a little bit about extracellular vesicles and all of the different lines of processing, so you first have both your production of extracellular vesicles, then EV isolation as well as characterization. You could say that microfluidics has a potential role for all of these. And so if we look first at extracellular vesicle production, there's all sorts of bioreactor design that can be improved with various fluidic handling and precise control. There's a nice review that's done here. And rather than going deep into EV production, because there's also been other talks, both in the education session talking about EV biogenesis, one of the things that I'd like to share, and I realize now my videos are not going to work, oh, maybe this one's going. So here we have a 1-micron capillary where we're forcing through cells that are going through the chamber. And so this would be for the extrusion for EV production. As you can see, the cells actually maintained their cell-cell connections. But you can start to see that there's actually a little bit of blebbing on the surface of these EVs. And if we take a look at these cells with some fluorescent dye, you can actually see both where the cells are lining up against one another, but you can also see some production of vesicles coming out. I don't want to name these vesicles because I don't want to be controversial, but they do contain cargo that's coming through. And if you think about some of the larger vesicles that we see, these are cancer cells that we're putting through our microfluidic device, that this could be one mechanism for EV generation as our cancer cells are traversing through some very tiny spaces in the body. But the most impact, I think, for microfluidics in EVs is thinking about EV isolation. Now, there are so many papers. There's actually I was just Googling it again, over 1,000 different papers now that are using microfluidics with EVs, with the majority of them being in EV isolation. Most of them are still early stage, where they're looking to see what can we learn and explore and how can we do this in our individual labs, but some are also scaling up quite nicely to then translate to the clinic. So I've highlighted some of them here, but I encourage all of you to do some searches in PubMed and to Google to see what these different types of technologies are. And I thought I'd give a little bit of an overview of our oldest EV isolation technology, which is using a herringbone chip to be able to pull out, where you put in your blood or your plasma, you enter it into our chip, there's one inlet, one exit. The size of the chip is the size of a microscope slide. Where we want to do that positive selection, we say we know what should be on the surface of these tumor-derived EVs. We want to be able to pull them out on our chip, and all of our unwanted agents continue out. So to talk a little bit about how this device works, or the different components that I was talking about that go into microfluidic device design, traditionally in a microfluidic device, you have laminar flow. So if you have your cells or EVs enter in, they stay in one streamline. They don't interact with the surface, which is where I'm putting all of my antibodies that I want to capture in my EVs. So what we do is add in herringbones into the surface of the device. And those herringbones are staggered and designed in such a way so that as your fluid goes through, it twists and turns, so all of the EVs interact with the surface, and we've modeled that to be over 80,000 interactions as the fluid is going through our device. So that's one part of why EVs capture onto our device. But then we also have to do all of that biochemistry onto the surface. So our group has now had 5 different publications of different biomaterials that we coat. We continue to evolve and change our biomaterials based on what we're trying to get. A lot of times we're just looking to make sure that we're anchoring our antibodies in the proper orientation to maximize our capture. So this is one biomaterial that we have that's thermally responsive. This was created by Eduardo Riategui, who's now a professor at Ohio State. But he had done this when he was a postdoc in my laboratory, where you simply raise the temperature to physiological temperature to release the EVs. We can also engineer the antibodies that we're anchoring onto the device so that it can help interact with the steric hindrance that would prevent these EVs from binding, and then compare it against other methods. So in the last little bit of time, let's see, trying to get my slides to advance. Here we go. So rather than, one of the things that we try to do, I'm gonna see if I can go back. Rather than thinking about just taking your microfluidics and saying, I'm gonna give you a chip, I'm gonna distribute these chips and all of the pumps, we can also use microfluidics to think about using the microfluidics to create the technology that we translate. And so Dr. Raheel Ahmad, who's a postdoc in my group, he's using microfluidics to create these, I don't know if any of you use Orbeez, if you have children, those gel beads that you can have that shrink and grow. So he's using microfluidics to create a microgel bead that's a replacement for, say, a magnetic particle to isolate out EVs. So he has created the system where it can create 30,000 of these beads a second. He then cross-links them, and this is just an SEM of what those beads look like. We can either use this particular surface property on these EVs to be able to isolate them, and he's now working on seeing if he can put antibodies against them. And these are those same beads after we've incubated it in bladder cancer patient urine. You can see the EVs are pseudo-colored with purple, so it's size agnostic in terms of the EVs that we're pulling out. And as he's working on this, we can see that there's actually an increase in yield when we compare this against size exclusion chromatography. And then we also are seeing a better purity when we're pulling them out by looking at TEM. So this is just another example, quick example of you don't have to send your microfluidics for that to be the product. You can use microfluidics to create your end product. And then, of course, there's all sorts of other ways to manipulate this, and so you can think about what other biophysical properties can you use, and how can you manipulate that with a microfluidic device. Viscoelastic sorting is something, also acoustic sorting are two big areas in microfluidics. There was really a lovely paper that came out last year in Science Advances that's looking at starting with whole blood and looking at how can we isolate out different EVs based on their size using these viscoelastic flows. And then finally, when we're thinking about characterization, there's all sorts of new ways that we can do single EV characterization. And so again, here's a review that highlights some of the different microfluidics that are there that you can look up on. Because I'm a mechanical engineer, I'm gonna highlight two that look at mechanical forces on EVs. Joanna Dahl has done a really nice study with some of my colleagues at Mass General, where she's looking at the biophysical property and how that's different in a particular type of glioblastoma. And then just last week, coming out, looking at the membrane fluctuations and surface adhesions of these nanovesicles out of the KTH. And this is just to show the more we learn, there's a synergistic combination of as technology improves, we can understand more of these different EV populations. Then we can go back to the fundamentals and theory of fluid dynamics to think about how can we create these new tools and technologies to both better understand, generate, and characterize these EVs. So I hope I've gotten some of you excited about microfluidics. At the end, you can always find me. I also like talking about microscopy, so if you don't like microfluidics, you can always come up and talk to me about microscopy. I'd like to thank all of my collaborators, our funding sources, but particularly my lab, and I'd love to take any questions if you guys have some.
Thank you very much, Shannon, for this great presentation. Are there questions? So while people are thinking of their question, maybe I can I can ask you. So you alluded to it, but the balance between having to run large volumes and the time it takes. So do you think that it's close to reaching a solution where people will use microfluidic routinely and not spending their whole day doing it?
Yes, so I think there is, and I think there's a question about what are the first questions you ask at one of the panels before? And so with the higher volumes versus long time periods. And so some of the devices like acoustofluidics often is pretty slow in terms of how long it takes. And I think it depends on the yield that you need to have. So for our technology that uses 2 liters of fluid, that's because you're looking for something that might only be present like 100 cells in 2 liters of fluid. So you need to have the large things. But you can then take the other alternative. So that's fast, it uses 2 liters, but is any patient really going to want to have to give that much volume when they come through? So there's lots of different considerations for that. Ideally, though, if you're thinking about bringing something to the FDA, you want something that's going to have an answer in 15 minutes. So you want to be able to divide whatever your volume is and your technology so that you can get an answer that quickly.
Thank you. So one question, Guillaume.
Yeah.
Guillaume Van Ile, France. Thank you for your talk, very beautiful. I was wondering what is the physical limit in the microfabrication? How small can be the channel that you produce? Can they be 100 nanometer width? Or?
Yeah, so if you use e-beam lithography, which is literally using electron beams to produce some of these devices, so you can get down to the nanometer scale. And so the smallest that we've been able to produce at the Harvard facility is 10 nanometers. But you can go very, very small. But again, with to Clotilde's, the time, if we're using e-beam lithography, it usually takes 24 hours to produce one device. So again, it's that trade-off of time versus resolution that you have.
Thank you.
Yeah.
One more question, yeah.
Thank you for this beautiful overview of microfluidics. I want to get back to original question on the trade-off between the precision of sample treatment and throughput and scale up. If we are looking not at individual patients, but let's say EVs as a manufacturing platform, when you need to work not with the liters, but maybe 100 liters, probably microfluidics scale up is perceived through multi-channel treatment. That's how the throughput is achieved. Your trade-off comes with manifolding, and especially if one or two, a few channels get clogged, or you mentioned bubbles formed, there is a flow maldistribution happens. Are there any solutions that are realistic and practical that can ensure solving this problem if we want to scale up and go to really biomanufacturing applications? Or is it going to be always a technology for the lab?
Sure. Great. So if I could just summarize the question, there is that challenge when you're trying to increase throughput, and it's always been, at least when I started doing microfluidics, if you want to have something go faster, well, you just make 100 of them in a row, and that's how you increase the throughput. I think we're getting better at that. We're not just making it be at 100 per row and looking at ways to be innovative. So our one that processes 2 liters of fluid, we actually put there's a magnetic core that we put inside the device, so we have this micro lens effect that's inside there so that you don't have to make 100 different channels to be able to go. And so thinking in fabrication innovation, so that helps a little bit with both creating the effect so that your flow rates can be higher. Bubble traps are something that's often included in devices when they're scaled up, so that if you have bubbles coming through, you can look at how to aggregate those. So there's always different kind of fabrication innovations that we're looking at to help increase that innovation level. I think it's something that with all technologies, we're still continuing to advance in how we do that, and there's still certainly room for improvement in there. But I would definitely say, like, on one of our devices, we have some bypasses so that if there is a clog, then it goes, and we have lots of redundancy inside our device so that it doesn't actually, because we consider every patient sample to be very precious, and so we don't want a device to clog and then us not to be able to process it, so we can engineer in bypasses. I hope that answers your question.
Thank you. Thank you very much, Shannon.
The last speaker of this session is Dr. Randy Carney from the University of California, Davis. He will tell us about EV heterogeneity and clinical applications.
Yeah, thank you very much, and I appreciate the invitation and the water. I was hoping for a beer, but water's okay. I guess that'll be in a little bit. So EV heterogeneity has definitely been a great theme of today, so maybe I can pull together a little bit more details and maybe some different ways to think about EV heterogeneity. And of course, if you just take the simple Google search of what EVs are by doing a quick image search for exosome or EV, you'll apparently see that they're all perfectly circular and unilamellar, They're about the same size as each other, and every single EV holds every single component of all the biomolecules that we know are actually trafficked by EVs in bulk. But of course, we know that this isn't true. We've heard a lot today about the different cellular and disease-associated factors which dictate a lot of the diversity of the EVs that we see. And nowadays, a lot of our cartoons are getting a lot better in terms of the different diversity of EV sizes, shapes that we can actually see. I would argue that our cartoons are still a long way from actually capturing what we're now sort of understanding to be the true nature of vesicles. We're getting better, so typically you see this old model, right, where kind of how I made fun of on the first slide, where everything seems to be packed in a single vesicle. In fact, now we know that many EVs are actually empty. They may only have a few proteins or nothing at all. And cryo-EM and other techniques are helping us actually visualize a lot of this density. I would still argue that these cartoons, even though it's excellent compared to what we had before, there's still inaccuracies which bother me as a former material scientist, which is like why is the membrane getting skinnier as the particle gets smaller, right? The thickness of the membrane and the size of the lipid should be the same. So I made an attempt to try to draw still a cartoon abstraction of EVs, but actually that are in proportion to each other. So this is ranging from about 30 nanometer to up to 150 nanometer cross-section, and you can actually get a better sense of the problem with stoichiometry here, which is that the smaller vesicles, and I would argue that 50 or 60 is actually the true mode, and I'll show you evidence for that from different sources, they hold very, very few molecules. You can see the intraluminal volume of a 30 nanometer vesicle is tiny compared to the volume of the membrane, in fact. And if you look at the number of proteins that you could carry, even if the entire volume was just protein, these are very small numbers. So we heard a lot about this from the excellent recent talk about engineering EVs, of how many can you actually pack inside of these things. Their relative surface area and relative volume over what we know of a pretty small range of a subset of vesicles from just 30 to 150 nanometers is quite staggering. We also heard in a previous talk in this section that it's true, the stoichiometry is such that each individual vesicle simply cannot hold a copy of every single molecule that we can detect in the bulk. And this has been quantified, especially for proteins and RNAs. But we're talking that even for the most abundant RNAs, 1 in 100 vesicles can actually carry even just a single copy or a handful of copies of these. So capturing the true diversity, I think, is a huge challenge. And certainly there's been technologies which are trying to meet these challenges. This was an excellent preprint. Actually, I was just checking today if this has actually been published. Maybe the authors are here at this meeting. But we see that even just using simple sort of sandwich ELISA-based test, people are already starting to think about quantification of the relative yield of different membrane proteins in terms of being more or less copies at the single EV level. Some work that we published a couple years ago was showing that even depending on which tetraspanin that you capture, if you want to do bead pull down, as has been mentioned for immunoaffinity several times today, when you're looking at the co-markers, which are actually maybe the tumor markers that you care about, it's highly dependent on exactly which subpopulation that you actually want to detect. So you can see in the top right here that if you're looking for CD24 or EpCAM or HER2, depending on if you pick a single tetraspanin, you'll either see those or you won't. So this again is an issue of the stoichiometry of the individual molecules that we actually care about detecting. Now, in addition to the heterogeneity of EVs, we have all these co-contaminants, and we heard great summaries today of all the different things that get co-isolated with EVs, depending on actually how you're choosing to isolate them. But when you look in the literature, even though new evidence is mounting, and this is very similar to the talk I gave a year ago, particularly this slide, and not much has changed. When I still read EV papers, they largely quote some, I would argue, highly arbitrary size definition, usually in the first sentence, right? You can say they're from 40 to 150 or 30 to 100. And even in the quiz this morning, I laughed to myself, because if you find a vesicle that's 29.9 nanometer, I think maybe you're special. Apparently that doesn't exist. And the mode is always quoted at 100 nanometer, but we know that this is an artifact of light scattering and the refractive index dependence on how we're actually quantifying size typically. Careful electron microscopy studies from many people in this room actually have shown that the true mode of most EVs in many sources is actually much smaller than you can detect with light scattering. So we're only seeing a fraction of those with flow cytometry, NTA, and other technologies. And that isn't to say there isn't a lot of value in these techniques, it's just we should all be aware of what we're actually measuring. So refractive index heterogeneity is something that I rarely see discussed, but this is a huge source of differences amongst EVs. We can actually model the refractive index of particles using Mie scattering, and this is one of the ways that we can get from light scattering on a flow cytometer into nominal size, and there's some excellent work, particularly by Josh Welsh and other people in the nanoflow cytometry community that appreciate this very well. When you start doing more advanced modeling, rather than considering them as solid spheres, but thinking about EVs actually as coarse shell structures, you can get even closer at turning scattering into true size, of course, still above the limit of what can actually be detected, since they don't scatter much light at the smaller size. But even these models, which are among the most advanced, are using scattering to go to size by typically assuming a single refractive index. But we know EVs do not have a single constant refractive index, and you can see this most apparently on cryo-EM studies. Here in these arrows, these are three apparently identically sized EVs if you look at their nominal diameter, but you can tell by the electron density of these images that some of these vesicles are trafficking a lot more materials than others. They would have very different refractive indices. And there's even been some work in the field to try to use complementary, orthogonal single particle techniques to measure the distribution of refractive index in particles, and it's highly variable. Multi-lamellar vesicles are another source that are rarely talked about, in my opinion. There's an excellent review called The Speculative Mechanisms for the Formation of Multi-Lamellar EVs, thinking that these aren't necessarily just an accident of isolation, but that these are purposeful or real, sort of EV subpopulations which might have different mechanisms, thinking about things like endosomal escape and uptake and targeting and tissue barrier penetration. These actually might have quite different properties in and of themselves. They're highly observed. If you look at any cryo-EM paper, you can see them all over the place. And contrary to the argument that these might be caused by high ultracentrifugation speeds, the formation of these irregularly shaped and multilamellar vesicles seem to be independent of the isolation source, as you can see by this nice study. So I always like to try to think about how many EVs are in a unit of blood, let's say. And so I took some values from the literature and tried to guess a sort of range given the sizes and relative concentrations of things like red blood cells and platelets, EVs, lipoprotein, protein aggregates, and came to a model that looks sort of like this. Of course, this is assuming that the mode is around 60 nanometer and is log distributed, which I think is probably pretty accurate. And you can see by some recently published data in the center here, if we're counting EVs, you can see the green bar in the middle is actually the ones that are small and are unilamellar and perfectly circular. And you see these are definitely a different subpopulation, which tend to be very small. And I was very happy today to see many images of electron microscopy and cryo that actually, if you look at these images, you can see quite clearly that the small unilamellar and more highly numerous EVs in our preps are actually these 50, 60 nanometer ones. And the larger ones tend to be these multilamellar ones. By the way, the figure I'm using for EV count is coming from Britta Bettend and some other work about doing some inter-lab flow cytometry comparisons, which are admittedly probably undercounting EVs due to the detection limit of most of the commercially available flow cytometers, especially ones used in that study, which started some time ago. If you assume that maybe they're 10 times higher, you start to see that the number of EVs actually in blood is quite staggering. So these aren't rare populations. And I just added this because the nice talk by Guillaume, I think, showed earlier today this image which sort of corroborates this, right? So you can actually see, and again, this is based on fluorescence detection, so you're probably not seeing the very smallest ones, but we have evidence that the number of EVs are quite staggering. And so what do you do with all this heterogeneity? I think one thing, as I mentioned earlier, is of course to try to use complementary orthogonal techniques to try to get at some ground truth where they're in agreement. And so commonly, and sort of MySev endorse, you might see some direct imaging, like negative stained transmission electron microscopy, TEM, maybe combined with some light scattering, like NTA. I would say, just based on my anecdotal evidence in the literature, that this is the most common sort of background characterization you'll see in the literature. But even in this image, which I would say is a quite nice quality negative stain TEM, of course, as you know from what I've said so far, I'm biased to think that EVs are actually much smaller in count than 100 or 200 nanometer, as you see in this image. So I just took this image and did some bandpass filtering and boosted the contrast, and you all of a sudden see a ton of small vesicular type particles actually jump out. And this is amongst that background. Maybe it's an artifact or maybe it's lipoprotein. I don't think we can say. But you can see a lot of these sort of imploded EVs, which is this artifact of fixation that we commonly see in negatively stained TEM. So even in apparent images, the way you collect the data, the contrast that you're looking at, the fields of view that you choose can highly impact the apparent size distribution. And of course, if you're trying to match it to NTA or some other complementary images, you might have issues. So it's not just doing multiple characterizations, but it's really critically thinking about limits of detection and using caution and calibration to actually try to get to some ground truth. There are many different technologies, and I won't belabor the point because we heard a lot about this today, but it is useful to think of them in terms of their approximate limit of detection and their ability to be calibrated using NIST traceable reference standards, which is something that I'm happy to see the field moving towards more and more. Another source of heterogeneity, which is definitely harder to assess, is actually the functional heterogeneity of EVs. So we know that ultimately why we want to use these EVs is because of their function. I would argue that even in the cancer diagnostics or diagnostic space, finding EVs that are actually contributing to the etiology of the disease are going to be the ones that are actually translating into the clinic. So not that they're correlative, but that they're even causative. So figuring out which subpopulations in functional studies are actually responsible for the mechanism of action, I think, is just a massive gap in the field. And we heard about that this morning a bit as well. But there are things that we can do. And so I would challenge each of you that are doing functional studies of EVs to actually think about what are the molecules within your EVs that are contributing to the function. And to try to dose your EVs according to the distribution of those molecules amongst their individual subpopulations, and to implement controls and single particle characterization tools to feed back into these. And this is a scheme that you could follow, and this is aggregated from a lot of pieces in the literature, which are more than a 15-minute talk can expand on the details of how you might properly design a study to actually standardize a functional assay. So for the last few slides, I wanted to share a little bit about where we're at in terms of heterogeneity especially, but more broadly in terms of the clinical applications of EVs. So last year, we published a study where we just tried to mine the publicly available data of clinical trials using the terms EVs or exosome. We were surprised to find that many of EVs, which are in sort of phase 1 and phase 2 clinical trials right now, in their public-facing records actually don't use the term exosome or extracellular vesicle within their entire clinical description. So, for example, Kodiak has two formulations which are advancing quite far, and they call them exosting and exo something else. I have it on the slide, and they actually don't use those terms. So we realized we needed to actually check press releases and other sources to try to mine all of this data. It turned out that we found about 471 records of people that are actually reporting what they're doing with clinical trials. Most of them were diagnostic. You can see nearly half. Of the rest, or a third of the total, were also companion diagnostics. So these are predicting, for example, which medications that a patient might respond to rather than detecting disease. Most of these studies, you could see this dramatic acceleration into the past couple of years as our field has exploded. And if you look within those, this is for the therapeutic trials. You can see that mostly these are focused on respiratory disease and cancer. The route of administration tends to be intravenous, and more than half of the sources of these trials were using mesenchymal or stromal stem cell populations. and engineered cells coming close behind. Remarkably, more than 20% of those clinical trials actually didn't even specify the source of stem cells that they were actually using for their study. And so there's a big gap in actually what people are reporting and how they're describing them. And so part of our angle of doing this meta-analysis of clinical trials was just to ascertain how many people are even thinking about the diversity or heterogeneity of EVs amongst their clinical trials. And we found almost no evidence that anyone was doing that. Of course, you don't write a lot in these clinical trial descriptions, so maybe internally they were. Similarly to therapeutics, you can break down the diagnostic and companion diagnostics, and perhaps unsurprisingly, they're largely focused on cancer, detecting cancer from blood, urine, and starting to get into some CSF as well. And these are largely based on lung and prostate cancer. And we heard the excellent talk about where we sort of are with the clinical diagnostic space from Carlos a few minutes ago. But back to heterogeneity, we could only find a handful of studies that even acknowledged any source of heterogeneity that might come from different cells. I think one of the trials out of the 471 actually did immunoaffinity to actually isolate a population of EVs according to a single marker. And so in my view, I think it's an essential milestone for as we start to translate our work into the clinic to actually consider that these aren't homogeneous preps, but that they're highly heterogeneous. And it's worth noting that EVs are, especially their clinical application, are essentially a brand new field. And so this is an image from a nice review from Ainsley Group that came out a couple years ago. You can see, although EVs were apparently reported in the literature several decades ago, as we all know, their application, you can tell by the timelines in the clinical trial data I just showed, have really only been in the past 10 years. And so we're very, very new. And there are some therapeutics which are already advancing through clinical trials, of course none which are FDA approved so far. And I think some people tend to overreact at this and think that somehow EVs aren't working or more needs to be done. And I would just argue that EVs are heterogeneous and complicated, and we've come a long way, and we're definitely going to get there. It's worth thinking, though, are EVs the right thing to use? So in terms of therapeutics and diagnostics, and we've heard some nice arguments today, that it depends on what you're trying to do. So perhaps LNPs are a better delivery system than EVs in some contexts. And so it's always good to think about what advantages EVs have themselves. And I won't go through this table, but you can certainly take a picture and look later, and I'm happy to argue with you about any of these. So the final slide, I would say there's some milestones of things that we can still address together. And I think a lot of people in this room are and will be contributing to these gaps in the field. And so of course we need to get better at vesicle characterization. Standardizing functional assays and refining our clinical trials are two topics I talked about. I think engineering EVs, especially from the bottom up and coming up with wholly artificial nanovesicles that can recapitulate what EVs can apparently do, is a big gap. So I would argue that until we can actually build them from the ground up and make them in a tailored way, we probably don't understand what they're actually doing. I think especially glycans and glycocalyx are a huge gap in the field that we're very bad at sort of understanding and characterizing. We heard a lot about EV biogenesis today, and I think engineering ways to actually manipulate cell pathways and have cells create EVs, subpopulations on demand is also a gap. And then dosing. Dosing is really hard. What do you dose by? The number, the concentration, a particular biomarker, the amount of lipids. Given the intense heterogeneity, it's really difficult to just even do a study where you compare EVs to liposomes. And so I don't think we're there yet on how to properly dose EVs, and it's certainly context specific. And then finally, what we're all doing here, I think, is just sharing all of our work, including our failures, and collaborating together is one way that we're all going to move forward. So that's it. I'm happy to take a few questions or discuss at the coffee break with you. Thank you.
Thank you, Dr. Carney. This presentation is open for discussion. While people think and come at the microphone, Randy, I have a couple of questions myself. So we heard in previous session that there is toxicity. And can you comment on this? Is it only dose dependent, or what are you aware of?
Yeah, I think Charles Lai mentioned this morning the sort of anecdotal evidence, and our lab certainly sees it as well, that at the volumes and concentrations that we seemingly need to inject into animal models to elicit some therapeutic response, especially with native unengineered EVs, you do just get acute toxicity that may be just related to the viscosity that you're delivering, or you can actually see animals almost like suffocating, even with EV doses of these. And it's just something that a lot of people talk about, but we haven't really seen it in the published literature. So I think, we've certainly had that experience, and engineering approaches like we heard about, in the previous talk in this session are going to help just get those numbers down. But yeah, and since we don't understand the mechanism of action in many of the unengineered EVs that we're applying, I think we don't understand the mechanisms of toxicity from the co-contaminants and other things which are coming along with these EVs.
Definitely. Thank you, Randy. There is a question there in the middle.
Hi, thank you very much for your presentation. I would have a question based on heterogeneity that we all observe and how little we actually know about what is all there in our preparations. What is your opinion? Are we there yet for standardization? Are the methods good enough to really standardization to make sense?
So are we there in even understanding the heterogeneity is the question? Yeah, I would say no, definitely not. And there's new papers that come out all the time which are unlocking new things that we're understanding. The tools are definitely getting better, and I think the characterization tools are advancing a lot quicker than isolation technologies. So right now we're sort of trapped in a position where, although we may be able to explain a lot of the heterogeneity by using advanced imaging, we saw a lot of excellent improvements to technologies today, like super resolution microscopy and force spectroscopy, this recent paper that Shannon was just talking about. But it's sort of inactionable to us right now because we don't have isolation technologies that can actually take advantage of this, getting stiffer EVs separated from less stiff EVs, these sort of things. So yeah, I think we're understanding heterogeneity better and better, and then hopefully the enrichment technologies that'll enable us to look at the functionality of these different subpopulations will be coming soon.
If I may, if you would need to pick something, one method, what would you suggest to actually provide the insight into the heterogeneity of the products?
Yeah, I always encourage people to learn and implement cryo-EM. I think it's one of the most powerful tools that we have for unbiased sizing of EVs. Unfortunately, it doesn't do well for concentration analysis. Super resolution. We have these new techniques which are enabling a more broad look at the total EVs. Nothing captures everything perfectly. So there's, try new tools that are available if you have access to them at your universities or companies. But in the end, there really isn't ever going to be a single tool that we're going to use that can capture the heterogeneity. So it's all about orthogonal, multiple techniques and trying to compare them, in my opinion.
Thank you.
Thank you.
Thank you very much, Dr. Carney.
Thank you.
Thank you, everybody.